Welcome back. In today's class, we will be learning about rhodophyta. The best example for your rhodophyta is Glaciaria. This is a red colored algae. There comes the name rhodophyta. Gracilaria is one of the most economical valued algae that comes under Rhodophyta members. First, we will understand the significant characteristics of Rhodophyta and then we'll learn about Gracilaria. They are nothing but the red algae. These algae are believed to be seen in the water bodies, especially in marine condition. They do not have any flagella. They are believed to be stuck with this rock, or they are stuck in with a solid substratum with the help of a modified discoid holdfast. They are photosynthetic in nature. A few species are freshwater living, mostly they are marine living. They have a complex or well developed reproduction. They are multicellular in nature. The thalli of the algae, all the organisms of red algae, are believed to be highly evolved, branched. They have a main axis, and the lateral branching can be differentiated very well. They're heterothallic in nature and they contain a set of pigments in them. One such very important pigment would be a phycoerythrin, which makes it red in color, and there are many accessory pigments accompanying it. They are believed to be saprophytic in nature or parasitic or epiphytic in nature. The cell wall is made up of cellulose and it has a reserved food material of starch. They show sexual reproduction by the production of gametes. They are generally developed oogamous type, where the female and male gametophyte would be completely unlike. First, coming to your Gracilaria, we see that it is a best example for your red algae. The best example for your red algae is your Gracilaria. This is the systemic classification of Gracilaria as we discussed. They belong to the family Rhodophyta and they are marine living organism. There are about 100 species of Gracilaria that are identified till now. They are favorable temperature would be in a little high temperature. They're seen in tropical sea waters. They are having a better developed thalli, where they have a main erect axis. They have a main axis, and then there comes lateral branching. We call them the first branching from the main axis is a first generation branch, and from the branching that occurs from the first generation branching is called as the secondary generation branching, and from that comes the third generation branching. The branching can be alternative, the branching can be dicotyledon. It can also be irregular at times, but the lateral branching are always attached directly or indirectly to the main axis. Thalli is believed to be thick, it's fleshy, it is cylindrical or terate. They have compressed or flattened thalli. This is how your exactly red algae would look like. They have a thick photosynthetic main axis which is fleshy and it has many lateral branching around it. The organism is not free living, it is attached to a suitable substratum with the help of a holdfast, a discoid holdfast. Sometimes, to show better evolution, it has secondary holdfast that comes from the margin of the branches. When we take the transverse section or the lateral section of the main axis, this is how they would look like. They have a cortex in the corner where the parenchymatic cells are smaller in shape and they are compactly arranged. And then comes the medulla, which is the middle part of the main axis where the parenchymatic cells are larger. Why we have to pay more attention for its main axis is this is the region which contributes to the sexual and asexual reproduction, be it any type of gametophyte synthesis or be it any type of spore synthesis. It all starts from the cortical region. This is the cortical region where you have the parenchymatic cells compactly arranged, and then comes the parenchymatic cells that are loosely arranged, that is medulla. This is the transverse section and the horizontal section of your lateral and horizontal section of your main axis, comprises of cortex and medulla. The middle region is your medulla. The superficial layer around is called as the tightly packed cells or the cortex. Cortex is a place generally where gametophytes are synthesized. To speak about the pigments present in this main axis, they have chromatophores, erythrophycoerythrin, it has chlorophyll A, it has chlorophyll B, it has alpha and beta carotenoids, and it also has certain reserve food materials like your starch. Coming to the reproduction of Glacolaria, it is a developed, modified organism, evolved organism. So it shows both sexual, asexual, and in fact also vegetative reproduction. Your vegetative reproduction happens by simple fragmentation, which does not involve any gametes, it does not involve any spores. But in case of your asexual reproduction and your sexual reproduction, there is involvement of spore production and gamete production respectively. These are believed to be oogametes type, as in the gametes produced by Glacolaria for the reproduction are genotypically, phenotypically completely unlike from each other. This is how your male gametophyte would look like. Your male gametophyte is called as the spermatangium or antheridium, where antheridium produces antheridia, or we can also call it as spermatangium produces spermatea. There are about 2 to 3 antheridia that every gametophyte would produce. And this is how your sporangium looks like, carposporangium looks like, which is the female gametophyte, which has a place or a habitat for the fertilization to happen. As and when your spermatangium produces 2 to 3 spermatea or your antheridia, it goes chemotatically and attaches to the female gametophyte. Your female gametophyte is simply divided into trichogyne and trichocarp, where your spermatangia will pierce through the ostiole, passes down all the way from your trichogyne and fertilizes the egg nuclei that is present inside the female gametophyte, that is your carpogonium. So your female gametophyte of your Glacolaria is called as carpogonium, which has the egg cell. Spermatangium is a male gametophyte that produces the spermatea, which is a male reproductive organ. Now that goes and dissolves the ostiole of your female gametophyte, passes down the lane from the trichogyne, and it fertilizes the egg that is present in the trichocomb. So this is how your position of your spermatangia would look like. Your spermatangia is scattered all throughout the surface of the thalli at times, or it cannot be interrupted, it is continuous, or it can also be present in the depressed cavity or a shallow cavity where your spermatangial conceptacles are present. Spermatangial initial cell will have spermatangial concept tails, and that concept tails will give rise to spermatea production. Or if not, spermatangia is also can be present deeper concept tails, devoid of long elliptical view in its structure. So that is also possible. These are the three main areas where your spermatangia will be produced in the thalli. Spermatangia is seen to be globular or ovule in shape, scattered all throughout the surface of the thalli. Coming to the female gamete or the female organ of this Glacolaria, it is called as the cystocarp. Your cystocarp or your carpogonium, firstly your carpogonium is divided as two important parts. The first one is the trichophore and the second one is the trichogyne. Dissolving the ostule, the spermatia that comes from the spermatangium dissolves the ostule and travels down all the way from the trichogyne and fertilizes the egg cell that is present in the trichophore. So once the fertilization of the egg happens, the egg cell starts developing, it becomes diploid in nature, the zygote starts developing, and then it produces the daughter cells. The diploid nuclei will now generally undergo meiosis and produce more nuclei. So instead of getting into the details of fertilization process, just now let's understand what are the organs present. The male reproductive structure is called as spermatia, which produces 2 to 3 spermatia or your antheridia. Female gametophyte is called as a cystocarp or your carposporangium, which has two important regions. The first one is your trichogyne and the trichophore. Trichogyne is a pathway through which your spermatia travels through and then fertilizes the egg that is present in the trichophore, and then it undergoes to be becoming a diploid zygote. Apart from that, it also has some sterile filaments which helps in the reproduction process. After the reproduction of your fertilization of the egg with the spermatia, here we have carposporophyte production. Carposporophyte production happens once the spermatia reaches the trichophore egg all the way from the trichogyne, developing a diploid zygote called as carposporophyte. So to understand the sexual reproduction of your Glacularia, the first important gametophyte production happens in the thalli. There would be female gametophyte, there would be male gametophyte. Female gametophyte will give rise to carpogonium as we studied. Male gametophyte will give rise to spermatangium as we studied in the previous slide. Now together, spermatia will go and fertilize the egg that is present in the trichophore. As we saw here, how the spermatangium produces spermatia and it helps to fertilize the egg present in the trichophore. It helps in fertilizing it and thereby producing a diploid zygote. Once your diploid zygote has been produced, it now develops, your carpogonium will now develop into a cystocarp. Your cystocarp will now produce cystocarpospores. These carpospores will, on set of suitable substratum and favorable condition, will start developing, and then they become a thalli. They become a young thalli producing tetraspores, as in they become tetrasporophyte. Tetrasporophytes are the type of thalli that produces tetraspores. Now this tetrasporangium will now produce tetraspores, where 4 spores will be produced, where 2 spores will germinate and become female gametophyte, and then 2 spores will produce and become male gametophyte. So this is how the cycle of Gracilaria works, where round the way they produce spores and they germinate. They either become gametophyte or they become sporophyte. This is why we say there is alternation of generation between the gametophytic stage and the sporophytic stage, where by the production of gametophyte, cystocarp is produced. Cystocarp will now produce a young thalli, that is a tetrasporophyte. Tetrasporophyte will produce tetrasporangium. Tetrasporangium will produce tetraspores. So in 4 spores, 2 spores will become female gametophyte and 2 spores will become male gametophyte. To study the structure of the cystocarp, the first one is the pericarp. It is a pigmented multi-layer compactly arranged parenchymatic cells. Then comes the gonymoblast. Gonymoblast is again present in the center of the cystocarp, which is made up of parenchymatic cells, and it has globular cells in that, which leads to the formation of carposporangium on the top. Always gonymoblast will have a carposporangium on top of it. This carposporangium only produces carpospores. These carpospores are round in shape, they are rich in protoplast, and they are non-motile, spherical in shape, they are triploid in nature. Later, they become tetraspores, and absorbing filaments are the sterile filaments that are present around, acts as a fencing for the cystocarp. Cystocarp is prominent, they are protruding, they are globulous, they are hemispherical, scattered all over the surface of the thalli, and these are the four important parts of the cystocarp. Cystocarp is believed to be a product of the fusion of the gametes in the gametophytic condition. This cystocarp will now produce carpospores. Those carpospores will start germinating and they produce tetrasporangium, and they produce tetraspores in turn. That is why we have this alternation of generation of gametophytic condition and sporophytic condition. Then comes, this is how your tetrasporangium or tetrasporophyte situated in the thalli. They are always situated in the cortex. As we studied the transverse section or the longitudinal section of the main axis, they are attached to the cortex of the cell. These cortex cells will now give rise to tetraspores. Tetrasporangium gives rise to tetraspores. It is situated in the cortex of the cell, where it has four tetraspores in it, where two becomes male gametophytes later, two female gametophytes later. So the first one is the isomorphic generation. Then in the gametophytic condition, we have the male cell gametophyte production, that is your spermatangia, and your female gametophyte called as carpogonium. And then there is a fusion of it. As a fusion or the result of your gametophytic fusion, cystocarp is produced. Now cystocarp will produce carpospores. These carpospores will produce tetrasporophyte. Tetrasporophyte will give tetraspores. These tetrasporophyte will give tetrasporangium. Tetrasporangium will give tetraspores. This is the order in which they undergo their life cycle, and there is alternation of generation between them. Without one, the other generation cannot be producing the offsprings. So these are the explanation for whatever was explained in detail. So this summarizes how your gametophyte will produce spermatangium and carpogonium, and how fertilization occurs. We know that as a result, cystocarp is produced. As carpospores are produced as a product of a cystocarp, will now develop as tetrasporophyte. Tetrasporophyte will give rise to tetrasporangium. Tetrasporangium will give rise to tetraspores, and these spores will now, on set of favorable condition, two will become male gametophyte and two will become female gametophyte. So that is all about your sexual reproduction. Apart from that, we have asexual reproduction, where two important types of spores are produced, that is carpospores and tetraspores. These carpospores and tetraspores are simply explained in this diagram, where we have spermatia and your egg cells being produced, or carpospores being produced. As the fusion of it, there is a production of carposporophyte. These carposporophyte will now give rise to carpospores. These carpospores, on set of favorable conditions, falling onto a suitable substrate, they become tetrasporophytic in nature. These tetrasporophytes will now produce tetraspores, where two will become male and two will become female. Just by the fusion of the gametes, there is sporophytic formation, sporophytic development. Here there are two types of sporophytes that are developed. One is the carposporophyte and the next one is the tetrasporophyte. Again, it goes alternatively. So this is how your carposporophyte and tetrasporophyte are produced. And concluding it with the economical importance of Glacelaria, they believe to be antibacterial, anti-inflammatory, antifungal, anticoagulant. There are many ways in which they are used for its flavonoids, for its proteins, for its pigments, for its lipid contents, for its proteinaceous nature, and its phenol content secreted by it. So this is all we have for today's class, where we started from First, we started from understanding what exactly are the general characteristics of Rhodophyceae members, and then what is the best example for Rhodophyceae gracilaria, its thalli structure, its appearance, and its main axis and pigment distribution. Then comes the reproduction of gracilaria, how different it is, and then how your sexual reproduction and your sporophytic and gametophytic shift occurs, alternation of generation is justified, and then we spoke about the asexual reproduction and then concluded it with the economical importance. So that is all we have for today's class. Thank you so much.